To welcome everyone and thanks for tuning in today. My name is Elliot Swartz. I'm the principal scientist for cultivated meat at the Good Food Institute. I'm joined today by Marie Gibbons from MG Consulting, and together we're excited to present the results from our new amino acid cost and supply chain analysis for cultivated meat. Today also marks the release of the full report. We'll drop a link to the download page in the chat where you can download the full report, the executive summary, and also the workbooks that contain the models and the data that we used for the analysis. What we'll do today and cover is just a brief introduction to GFI and also some background context and motivation for this work. I'll walk through some of the key sort of methods and scenarios that we use to build the model. I'll then turn it over to Marie to discuss the key results, and then I'll wrap up with conclusions, recommendations, and future directions. And we should have plenty of time for Q&A at the end. Just a reminder, you're welcome to drop questions in the Q&A box throughout, and we'll get to those at the end. All right, so for those that are unfamiliar, the Good Food Institute, or GFI, is a 501c3 nonprofit organization. We're a science-driven think tank that's helping to build a more sustainable, secure, and just protein supply. We do work across science and technology, corporate engagement, and policy that really sort of collectively aims to catalyze an innovation ecosystem that will advance the science and also market success of alternative proteins. GFI is actually a global network of nonprofit organizations, and we all operate under the same mission. About half of our global employees are based in the United States, and the other half are spread across teams in Brazil, Europe, Israel, India, the Asia-Pacific region, based out of Singapore, and most recently, Japan. All of our work is powered entirely by philanthropic support. So if you like what we're doing and you find value in the resources that we create, please do consider supporting us. So just to sort of start with, I wanted to discuss why are amino acids important for cultivated meat in the first place. And as it turns out, the bulk of cellular biomass in proliferating cells in a cultivated meat process is actually made up of amino acids. And so they serve as really a primary input into any cultivated meat process. Additionally, previous work had identified amino acids as a cost driver of cultivated meat production. So most notably, a report by David Humbird back in 2021 suggested that amino acids alone, indicated by this line item here, could cost anywhere between $18 to $19 per kilogram of cultivated meat production. In some scenarios, this is actually up to 50% of the total cost of production. And it also implies that essentially amino acids alone would push cost competitive cultivated meat out of the realm of possibility. If we look at today's amino acid supply chain, it's well understood that the amino acids that are used in media come from individual fermentation processes. And the suppliers for amino acids are based primarily in Asia, with a heavy concentration in China. And only some amino acids today are produced at very large scales, mostly the ones that are used for animal feed supplementation. Additionally, media suppliers serve as really critical integrators along the supply chain. And so individual media suppliers will have a network of up to dozens of individual suppliers of amino acids and other inputs spread around the world that they source internally. They bring those inputs in-house and they mix and formulate them together according to the needs of their end users, in this case, the cultivated meat companies. And so it's been proposed that one way to potentially get around this perhaps overly costly and complex supply chain of individually fermented amino acids is to perhaps rely on hydrolysates from plants, microalgae, agricultural byproducts, maybe other organisms, and actually use these as a primary source of amino acids. And so, for instance, in David Humbird's analysis, he looked at a soybean hydrolysate, which contains a pretty favorable amino acid profile that's comparable to the average amino acid profile of proteins in your cells. And if we could source this at around $2 per kilogram, then the cost contribution for amino acids to cultivated meat would be dramatically reduced. And so sort of taking all of this previous information together, we wanted to understand, what is the current situation with respect to the supply chain today for amino acids? What are the costs? What are the expected volumes? And are hydrolysates going to be an important future part of the amino acid supply chain? And so before I begin, I wanted to thank the people, in addition to Marie, who helped support this work. In particular, Amanda, who's our program manager at GFI, who also helped with some of the analysis. And none of this work really would have been possible without a broad set of industry stakeholders who provided information and insights to us that allowed us to sort of formulate and have high confidence in a lot of the numbers that we use in the model. So thank you very much to all of those listed here on this screen who participated, as well as several others who wish to remain anonymous. All right, so our analysis had a few key questions that we wanted to try to answer. The first on the volume side of things was how many metric tons of total and also individual amino acids would be needed to meet the demand of a future scaled cultivated meat industry. We sort of arbitrarily set this at 250,000 metric tons per year, which represents probably around at least 500 times larger than the industry is at its current stage. We also wanted to know what the anticipated cost contribution of amino acids were per liter of media and also per kilogram of cultivated meat. And how does this compare to prior estimates, such as the ones that I showed you on some previous slides? And on the hydrolysate side of things, which raw materials could be the most promising candidates for supplying amino acids? And importantly, what would they need to cost to be competitive with those from fermentation or the current supply chain? And so what we did was take existing public data together with structured interviews with the stakeholders that took place beforehand, and we combined all of this information to come up with some key sort of methods and scenarios that we use throughout the report. So the first key scenario is the bioconversion efficiency, or essentially what amount of amino acids is actually needed to produce 1 kilogram of cultivated meat. We sourced different numbers from existing published studies and supplemented those with some of the information that we gathered during the interviews. And we came up with a range of around 200 to 650 grams of amino acids per kilogram of cultivated meat production. And so 200 represents the most efficient scenario, whereas 650 represents the least efficient scenario. We also needed to understand what the concentrations of individual amino acids were in media that's expected to be used in commercial production. And a lot of people rely on DMMF12 for their modeling work. This is a published formulation that a lot of people use as sort of a basic basal media formulation. But as you can see, it's actually kind of nutritionally deficient compared to what is needed to support the demand of high density cell cultures. And so to represent media that we think is more commercially relevant and nutritionally dense, we first used the media formulation called FSS4. This was previously developed by Sci-Fi Foods, which is a former cultivated meat startup company. And together with some cell lines, GFI acquired the media formulation and the cell lines. Hopefully some of you have seen that news. And that media formulation is published on our website and also available in the full report, open access. You can see also that the concentration of total amino acids in the media is similar to that published by Believer Meats in a paper last year, which suggests that this sort of nutritional density would be expected in commercial production. However, if this wasn't the case, we also modified a CHO media that was available via a patent. And we use these two media formulations to sort of represent the range and perhaps potential differences in amino acid concentrations across media formulations that would be expected in the industry. And so you'll see in a lot of the results that Marie will walk through the FSF4 and modified CHO media featured in the figures. Lastly, the other piece of information that we wanted to assemble was actual prices of food-grade amino acids that are being used in the industry today. And so we were actually able to acquire real purchase quotes for less than $100 kilogram quantities of food-grade amino acids. This enabled us to establish a range from low to high for food grade price scenarios for each individual amino acid. And we also supplemented this with a third price scenario for feed grade prices that were sourced from websites like Alibaba. We know that people are testing feed grade inputs today in their media. However, this does pose some potential regulatory issues. And so really, at least for the purposes of this analysis, these are intended to represent more of a lower boundary for amino acid prices. So we combined all of these sorts of information to create a model that output several results, and I'll turn it over to Marie to walk through those.
Awesome. Thank you so much, Elliot. So we began by wanting to take a look at the projected volumes and supply of amino acids. So we did this by first calculating the estimated total, and that we found that we would need anywhere between 50 and 163 kilotons per year of total amino acids in order to produce around 250 kilotons of cultivated meat per year. And you can see in the chart here that we have a variety of different estimates ranging from 200 to 650 grams of amino acid per kilogram of cultivated meat, which of course had an influence on the estimated amount, and that's just due to differences in bioconversion efficiency. So next, we calculated the projected volumes of the individual amino acids. And we found that arginine, glutamine, and serine all had the highest projected demand, with these volumes often exceeding over 10,000 metric tons annually. Of course, this did depend on the media formulation and the bioconversion efficiency, and you can see these variations in the chart here, where we have on the y-axis the list of essential amino acids that we focused on. With our x-axis, we're looking at tons of amino acids, and then with our blue bars, we're focusing on a variety of bioconversion efficiencies within our FSF4 media. And then with our red X's, we're looking at the modified CHO media, but that's only at the very highest bioconversion efficiency. We just wanted to make sure that we weren't underestimating any projected needs of amino acids. And other amino acids can, of course, be seen in our full report, but we wanted to show some of the highest projected amounts here. So next, we wanted to determine how these volumes compared to current global production estimates of individual amino acids. So we found that, unfortunately, the future cultivated meat industry's demand for asparagine, glutamine, histidine, proline, serine, and to an extent tyrosine, isoleucine, and leucine could potentially outstrip the current global supply for these individual amino acids. Now, I do want to point out that the estimated yearly production of amino acids is incredibly difficult for us to verify. So we were able to get some of these sources, but future research is needed in order to make sure that these are super duper accurate. But these are the numbers that we have at the moment, even if they are, considered at lower quality than other numbers in the report. And we do think that special attention really should be paid to these amino acids as the production capacity for cultivated meat increases to 250 kilotons per year and beyond. So after looking at production volumes, we wanted to look at the costs of these amino acids and how those costs contribute to the media and cultivated meat production costs. So I'm happy to share that we found that food grade amino acids cost up to 10 times less than prices assumed in previous analyses. So in the graph here, we can see on our y-axis that we have all of the amino acids that we looked into, and our x-axis is the cost per kilogram of amino acid. And we're looking at several different price scenarios, with the blue bars being associated with the low and high food grade pricing scenarios. Our dark blue circles are feed grade, very low pricing scenarios. And then we have our red X's, which represent the previous analysis by Humbird. And this previous price estimate is more representative of pharma grade prices, whereas the food grade prices, which are based on real world quotes and often for purchases less than 100 kilograms, which is very promising because one would assume that as demand increases to over 100 kilograms, the prices might actually go down. But we found that even at this low amount, the amino acids tend to be between $5 to $50 per kilogram for most of these amino acids. So we wanted to ask ourselves, how do these prices contribute to the dollar per liter media estimates? So we then found that the amino acid cost contributions to media ranges anywhere from 2 cents to 17 cents per liter. And this is, of course, depending on the media formulation and the price of the amino acids. In the graph here on our x-axis, we're looking at several different pricing scenarios again, ranging from feed to low food grade to high food grade. And we did also include a Humbird price scenario. And then on our y-axis, we're looking at the cost per liter ranging from 0 to 60 cents. And we looked at two different media formulations, our FSF4 and our modified CHO. And we found that total media costs under 20 cents per liter are in fact achievable. And that's really exciting because it aligns with recent claims made by companies in the sector. So it's really nice to see some of these numbers verified independently. So we next wanted to figure out how these prices contribute to the dollar per kilogram cultivated meat estimates. And what we found here is that amino acid cost contributions to cultivated meat ranges anywhere from $1.19 to $15.22 per kilogram for cultivated meat. And this is looking at FSF4 media across three different pricing scenarios and several different bioconversion efficiencies, which are outlined in your y-axis. And then, of course, your x-axis again has your dollars per kilogram of cultivated meat. And again, this was really exciting because we found that by combining lower prices with more efficient bioconversion efficiency production, it does suggest that amino acids could cost fall or it could cost below $5 per kilogram of cultivated meat, which is represented in our red line here. And that again is a major improvement over previous estimates. So we next wanted to look at what individual amino acids were driving the cost. And here we found that arginine, asparagine, histidine, serine, and at times glutamine were all individual cost drivers. And this is based off of two media formulations and three different pricing scenarios. The three pricing scenarios are seen on your x-axis, and the different colors are associated with the different amino acids and how much they contribute to the cost of media. And one of note is serine, which stood out, accounting anywhere from 16 to 38% of total amino acid costs. And again, this did depend on the pricing scenario and the media formulation, but still very much something to keep in mind moving forward. So after looking at the production estimates and the costs of amino acids, we wanted to expand our horizons and see if we could find additional resources that could supply our amino acids outside of fermentation-based techniques. And this led us to hydrolysates. And here we went ahead and assembled the amino acid compositions of 50 different raw materials and commercial peptones and hydrolysates that were available to us. And we then compared them to the amino acids found in FSF4 media. So the heat map here shows the ratios. You can certainly get into more detail within the full report, but a ratio of 1 or greater indicates that 1 gram of the material meets or exceeds the concentration of 1 liter of FSF4 media. And so the greater the ratio, the deeper blue the color within this heat map here. And what we found was that, unfortunately, there wasn't a single material that could meet the entire nutritional demand without needing to add impractical amounts into the media formulation. And so moving forward, we decided that our models needed to assume that fermentation-based glutamine, arginine, asparagine, cysteine, and serine were all spiked in individually when needed rather than fully supplied by hydrolysates. So we then used ChatGPT, which everybody calm down. Whenever we used AI, we did replicate the findings using Excel's solver add-in, and then we definitely confirmed it manually with manual calculations. So any hallucinations were confirmed to be real. But we did use AI to identify candidate blends, looking at a combination of up to 3 different hydrolysates in hopes that they would satisfy the amino acid requirements of our media formulation. In doing so, we also looked at 4 different scenarios to see how potential blends would differ between media formulations and material data sets. So for our first scenario, scenario A, we looked at our FSF4 media formulation, And this was at a bioconversion efficiency of 350 grams of amino acid per kilogram of cultivated meat. We did initially consider looking at different bioconversion efficiencies, but we quickly realized that while the amount of hydrolysates would change as the bioconversion efficiency changed, the ratios wouldn't change. And so we felt pretty confident that just focusing on one bioconversion efficiency would be very reflective of all bioconversion efficiencies. Next, we also wanted to look at the difference between media formulations. So we looked at our modified CHO media formulation, again with the same 350 gram per amino acid per cultivated meat bioconversion efficiency. And then for scenario C and D, we wanted to look at what would happen when we restricted our inputs. So we would, for scenario C, only look at commercial hydrolysates, and then for scenario D, only raw materials. And the reasoning for that is unprocessed raw materials tend to have lower amino acid concentrations per gram of dry mass. So moving on, I want to walk you through what we're looking at here. So if you draw your attention to scenario A, we have blend 1, 2, and 3. And what we're looking at, for example, blend 1, we have a total of 449 grams of dry mass, which is made up of 3 different hydrolysates, and they include potato peptone, pea peptone, and yeast extract. And we applied this analysis across these 4 scenarios and identified our top 3 blends here. And so 449 reflects the amount of dry mass in grams of the total hydrolysate blend. And what we found was that potato peptone and pea peptone actually stood out as really good candidates for supplying amino acids in both of our media formulations. And interestingly, in scenario B, where we looked at our modified CHO media, we saw that it did require more material, which makes sense because there are more amino acids required for our modified CHO media. But interestingly, it also had more of a variation of materials, which is likely due to our higher proline requirements found in our modified CHO media, which could be addressed by including things such as wheat gluten, wheat peptone, and a specific type of yeast. And before we move on, I do want to again remind everyone that each of these scenarios assumes that glutamine, arginine, asparagine, cysteine, and serine are all individually spiked in. And while we are excited about these results and really hope that people will run with them and test them out, they are to be viewed as guiding examples. Several limitations do exist that we'll talk about in just a moment. But again, we are excited about it, and we hope that others will find additional blends that they can use. So moving on to scenario C and D, with scenario C, which limits our inputs to commercial hydrolysates, we found that they actually largely replicated the results from scenario A. And then with scenario D, where we limited the inputs to raw materials only, we found that we did have a greater diversity of materials, and we also required more material, likely because the amino acid content in unhydrolyzed material is a bit lower. So next, I want to walk you guys through a great example of our favorite hydrolysate blend, and it shows how 3 different hydrolysates can complement each other in order to provide the amino acids needed in our FSF4 media formulation. So as you can see here, with each individual amino acid, you can see how the potato peptone, the pea peptone, and the yeast extract are all able to complement each other and contribute to the amino acid needs. Of course, as we mentioned previously, there are a few amino acids that weren't able to be supplied by the hydrolysates and are spiked in, but otherwise, we were able to get a really great mix of amino acids needed for our FSF4 media formulation, specifically at our 350 grams of amino acid per kilogram of cultivated meat bioconversion efficiency. And also, our downloadable workbook has embedded calculators. So if this is something of interest to you, you can derive your own hydrolysate blends and use other data sets and scenarios to find something that works really well for your process and for your cell lines. So after looking into this, we wanted to determine the hydrolysate pricing windows that the industry could aim to target. And so what we did here is that we calculated what each candidate blend of hydrolysates would need to cost in order to be at parity with media that's made only out of fermentation-based amino acids. And these costs were based not only on the total mass of the blend needed, but also included the cost of the of the amino acids that were spiked in. And we found that that ranged from $1.51 to $11.27 per kilogram. And so that just provides the industry with a really good target pricing window for hydrolysate development. And speaking of hydrolysate development, uh, while hydrolysates are really exciting and promising, we did find in our interviews that there are several challenges that still need to be addressed, and these include things such as batch-to-batch variability, solubility issues, elevated ammonia concentrations, extensive optimization needed for each hydrolysate that people choose to use, limited compositional data, possible anti-nutritional factors, and potential increased regulatory burden since these are not chemically defined formulations and there could be potential allergens present. So while there is a lot of enthusiasm for hydrolysates and there is current testing going on within R&D programs, we expect that near-term cultivated meat products are unlikely to use hydrolysates for amino acid supplementation until we're able to resolve these challenges. So in summary, we found that serine, glutamine, asparagine, histidine, proline, and arginine were all high-risk amino acids, and these are due to a variety of factors such as high inclusion rates in the media, elevated prices, low production volumes, manufacturing complexity, and poor candidacy for the hydrolysate replacement. These six amino acids definitely produce or present opportunities for further innovation and focus, and if folks want to drop the costs of production. And for anyone who's interested in learning more about these high-risk amino acids, you can turn to our full report where we've provided snapshots of each of these high-risk amino acids, where we talk a little bit more about why they're important, what functions they serve, how they are posing a risk to the current industry, and opportunities where we can address these risks. And now I'll hand it back over to Elliot to go over our exciting conclusions.
All right. Yeah, thank you, Marie. That was a really great walkthrough of the key data and results from the study. I'll end on a few conclusions and then a couple of recommendations that we have based on the research. I think there are really a couple of key takeaways from the work that we produced. The first is that, importantly, really food-grade amino acid costs are significantly lower than what has been previously estimated in prior studies. So this is really, I think, the first study that tried to take a look at uncovering what the real costs of a food-grade supply chain that is being used today actually look like. And this, of course, corresponds to a much more optimistic outlook for cost competitive cultivated meat. And so obviously this doesn't solve all the challenges with respect to scale up and cost competitive production, but it does provide a little bit more optimism and realism toward what the industry is working with today. Additionally, as Marie showed, as the cultivated meat industry scales, it is possible that the demand for certain amino acids could actually strain the global supply. And so we think it will be important for suppliers and other actors along the supply chain to really proactively plan different capacity that might be needed for different amino acids that will help prevent bottlenecks from a supply standpoint and also price shocks if demand suddenly does start to outpace supply in the future. And then lastly, hydrolysates, which are being used, as Marie mentioned, in a lot of R&D work today, we think really hold a lot of long-term potential to be incorporated into the supply chain, but there is still a lot of work that needs to be done to figure out, how are we going to process these materials, which materials are going to be used, et cetera. And so there's, probably still some time between now and when they're more routinely used as basal media nutrient supplies. However, there has been a lot of progress made on using hydrolysates for replacing different proteins in the media, and a lot of published work on that already. So hydrolysates could be used in different forms and formats for media inputs, but as a basal media supply or replacement, still quite a bit of work to do, likely. All right, so some recommendations and future directions for different stakeholders who are involved in cultivated meat production and the supply chain. The first is we touched on this sort of concept of proactive planning or ensuring industry readiness. So one of the things that sort of came up in the interviews and people talk about when it comes to scaling supply chains is this concept of pooled procurement, where essentially all of the companies are using the same amino acids to some degree. And especially for the ones that are of highest risk, what can actually be done is pooling the demand from all of the companies in the industry to signal a stronger demand from the industry's standpoint to the suppliers so that they can really be incentivized to build out new capacity or production lines for these amino acids so it's ready when the time actually is needed. Additionally, governments and policymakers have a role to play here, especially governments who are looking to take a leadership role in the cultivated meat industry. They can incentivize onshoring of amino acid supply by offering things like grants or tax incentives and low interest loans to amino acid suppliers. This can stimulate local job creation, enable countries to leverage local feedstocks that would feed into the fermentation processes, and also ultimately mitigate risk of supply chain consolidation under the umbrella or roof of a single entity. For the development of hydrolysates, as we mentioned, we know that a lot of people, both in academia and in startups, are testing these out. And so we do hope that the strategy and also price targets that we show in this analysis will be helpful for you to not only sort of have a target price for developing cost-effective hydrolysate processes, but also narrowing the search space for testing that will hopefully lead to more sort of hypothesis-driven research and increase the hit-to-lead ratio that you find when you're screening different materials. Additionally, as we talk about in the report more, there's a lot of sort of complex challenges and interactions when you begin to consider using hydrolysates as basal media replacements. And so we think what will be important is really being able to create open access data sets, not only on the composition of different raw materials from different processes, but also how they perform in different cell lines, species, and also just contexts overall, whether it's in 2D or 3D culture, for instance. And so having these data sets available, we think would be really a prime use for AI and machine learning techniques that can maybe sift through this data, develop new hypotheses, and ultimately, enable you to sort through this complex challenge in a little bit more of an efficient manner. One of the other things that came up that we didn't discuss in this presentation, but is included in the report in several sections, is the importance of the regulation of media inputs and the grade at which they're produced. And what we uncovered through the interviews is a lot of sort of optimism that we could perhaps develop a fit-for-purpose cultivated meat grade set of specifications that essentially combine what you need from a cost perspective, from a safety perspective, and from a cell performance perspective that will be suitable for use in cultivated meat rather than relying on grades as they're currently defined, which sort of sit maybe in between what is needed for a cultivated meat industry. And so ultimately, this is something that would require a lot of collaboration across not only the companies and suppliers, but also the regulatory agencies and other experts to sort of develop what the set of specifications might actually look for some of these key parameters here. And ultimately, we think that this could be a way to reduce costs, ensure that everyone's operating under the same set of expectations, and also streamline the regulatory review process in the future. Additionally, we discuss, the need to close or further sort of narrow in on some of the data that we identified or use in the report. And so one of those key data gaps is around the actual bioconversion efficiency for amino acids. You can think of this as a greater part of the ultimate feed conversion of the overall process. But essentially, the feed conversion ratio has an enormous impact on the overall cost and also environmental impact of production. And so this range is actually still, fairly wide. And I would like to see it be narrowed down with more empirical data from real world processes. And so I leave that up to the scientists and some of the startups to really publish some of this empirical data that will help us narrow in on provide a narrower range for future studies that will improve the accuracy of cost and environmental impact models in the future. As Marie mentioned, the data for production volumes of amino acids that are suitable for cultivated meat are somewhat difficult to find, although we didn't necessarily exhaust every potential option. But we do think that, ultimately we're going to need some more reliable data so that we can really ensure that, the amino acids that we're identifying as potential bottlenecks are indeed the ones that we should focus on in the future. Some people can perhaps let us know if you have that information on hand afterwards. And then finally, as many people are probably wondering is, if you look at the cultivated meat industry as a whole, they use a variety of different cell types, different species, different processes. This all implies that there's going to be a variety of media formulations. Basically, every media formulation in the different processes is going to be customized to some extent. And so the ones that we use, the two in our report, we think are commercially relevant, but that doesn't necessarily mean that it encompasses all of the necessary, potential scenarios of production. And so the only way to really find out the degree of variance is to have more open access media formulations available for the community. Again, this is something that companies can do, scientists can work on in the peer-reviewed literature, and also potentially nonprofit actors could acquire IP from startups if they go under. Lastly, environmental impacts. I do want to mention that, one of the sort of consequences of our work is that we think that, the industry, at least for the foreseeable future, the next few years, is likely to predominantly rely on amino acids from fermentation. This actually runs counter to some of the LCA work that's been published, including work that GFI funded and collaborated on with CE Delft, where we assumed in that study that 75% of the amino acids would be supplied by hydrolysates, which is, probably not going to be true by the year 2030, at least. And so what this means is that it could have consequences on upstream energy use in the supply chain, water use, land use, et cetera, depending on, the feedstock that's used in fermentation processes. And so a big piece of missing data that will need to be filled in is really getting food grade amino acid production from real world processes to be entered into environmental databases that life cycle assessment scientists use to perform these impact analyses. Right now, the available information in these databases is either outdated or in some cases non-existent. And so I think it will require really scientists to collaborate with the actual suppliers in the sector to uncover this data, ensure that we have accurate information available in these databases. All right, that concludes the webinar. Thanks everyone for your attention and listening. I hope you'll take the chance in the next few weeks over the holidays to read through the full report and get back to us with feedback. We'd love to hear it. And for now, we can stop and take any questions. So one of the first questions that was asked was around, do most of the assumptions for amino acid bioconversion into kilograms of cultivated meat account for 100% cell inclusion in meat? Or does this include things like hybrid products? So I can answer that, and Marie, please feel free to chime in. Essentially, we assume that the kilogram of cultivated meat represents 100% of wet mass of a cultivated meat product. We actually do not specify the water content that's assumed, because a lot of the bioconversion efficiency data that we derive from in public in studies, it actually varies in terms of the water content assumptions that we made. And so that's also part of the reason why we use a fairly wide range in this analysis, is to sort of basically account for differences in water content assumption for different products. Most people assume 70 to 75% water for conventional meat products, but in reality, cultivated meat might have a higher water content, which ultimately would probably require fewer materials to create a kilogram of cultivated meat. Nevertheless, yeah, this gets a little complicated, but the range that we use is supposed to account for that. And presumably, if you're creating a hybrid product with only a 50% inclusion rate of cells, then you can split those numbers in half, for instance. Okay, there's another question on the bioconversion efficiency. I think I just answered this, but I'll read the question as well. So if they wanted clarification on the bioconversion efficiency range and whether we considered dry weight for amino acids and wet weight for meat, yes, that is what we did. And as I mentioned, we assumed a 100% cultivated meat product, and that is on a wet weight basis. Those details are included in the extended methods section of the report, by the way. All right, Marie, I'll let you answer this question here.
Yeah,
Where
great.
can we find the updated costs for amino acids? Are they available for us to use these values?
Yes, so we can't provide you the sourcing of our amino acid costs. We got those from confidential interviews, but we were able to publish them in the report. So you can definitely go to the report and get the most updated amino acid costs, and then use those as a baseline when you're negotiating prices or providing amino acids.
Yeah, and yeah. So one of the other questions that's sort of related to this on amino acid costs was asked is when amino acid costs are reported for the media, the focus appears to be raw material costs. Was consideration given to many non-zero costs needed to maintain high quality supply chains over time, teams of sourcing, FSQR, management of inventory, QA, QC, et cetera? Yeah, this is a good point. So the costs that we sourced were like from a direct purchase of amino acids. And so obviously, if you're a media company, you're doing a lot of providing a lot of value to ensuring that these inputs are well maintained. They have a documented supply chain. Their quality has been assured, et cetera. And that obviously is not free. And so we think we didn't get into the types of costs that those add to each individual amino acid or component along the supply chain, as well as the types of margins that media companies are seeking or are desirable, whether current or in the future. That's probably work that needs to be done if we want to create even more accurate models from a supply chain standpoint. We just didn't really have the time in the scope of this analysis to really uncover all of those details. If you want to share them with us after, we'd be happy to learn more, just so we're aware. But one of the things that the reason why hopefully this isn't much of an issue is that what we did was take the amino acid costs information that we received, which again represents sort of direct purchases, and we established a range, which is essentially kind of like a plus or minus 25% off the midpoint of what was the highest or lowest value that we receive. So hopefully within that range is still representative of the, let's say, markup that would include these costs. But yeah, that's something that is an important piece of nuance that has to be in consideration, I think, for how these data are interpreted. And we do have, I'll mention in the extended methods, there several limitations to some of the methods, let's say, that we use to analyze this information. And so there's a whole table in the extended methods of the report that walk through some of the limitations to ensure that people are interpreting the data correctly. I will take this question, Marie, that I highlighted, and then I'll jump over to you. So someone asked, I'm not sure if this was answered already, but which cell types were used to model the data? Do you expect the amino acid requirements to vary between cell types? Yeah, so I hinted at this, I think, in one of the last summary slides, but really this is intended to be agnostic. So there's no specific cell type that this is intended to be most accurate for. The FSF4 media that we used was designed to support bovine fibroblasts in suspension, and obviously the CHO media was designed for CHO cells and slightly modified based on what we know about cultivated meat production and amino acid demand. So we think, a lot of animal cell metabolism has similar requirements. However, there are obviously differences across species and cell types, and so there is variance there that is to be expected. And so this is, sort of a first shot on goal that gives a pretty, hopefully, good high level overview of what is to be expected. But again, in order to really understand the degree of variance across different cell types and species, we're just going to need a little bit more public media formulations available. I don't know if you want to add anything to that, Marie.
No,
No.
nothing to add there. Great job, Elliot.
Okay. So Marie, do you want to address this question,
Yes.
which which
Oh,
is you can read it. Yeah, go.
yeah. So someone asked what downstream processing needs are needed to post hydrolysis steps typically. So that is very dependent upon the different material that's being hydrolyzed, as well as the target results of what you want out of the hydrolysis process. But when we dived into the literature and dove into the literature and spoke with folks in interviews, it sounded like very typical methods involved some sort of solvent extraction, potentially an enzymatic process. Some different hydrolysates could actually just be treated with heat, and then they'd be good to go. Some folks were exploring sonification. Filtration was also a really popular method. So this is something that I think is really important for us to dive deeper into, because it is a big cost driver of hydrolysis. And again, it varies from material to material. So great question. I wish I had a better answer for you, but at the moment it's lots of different things.
Yeah, and I'll touch on this other question that also asks a little bit about hydrolysates. But first, someone asked, for amino acids whose supply chains may become volume limiting, did you query with existing manufacturers whether the current production method is easily scalable if the market demand justifies it? So this is a good question, and this is sort of what I would view as a next step in terms of understanding. So we really scratched the surface, did a little bit of research to understand, which amino acid production processes are maybe more complex than others, and we talk about this a little bit in the snapshots of the report. But really, because these are individual processes, they all have their own like quirks and information associated with them. And so it's kind of like follow-on work, I think, that would need to be done to really investigate that question on, scalability, whether new innovations are needed. So the purpose of our report, or at least where we got to, is to really identify the ones that should be the focal points. And so those are the ones that, perhaps offer the opportunities for cost lowering innovations on the manufacturing side of things, perhaps using different feedstocks, that sort of thing. And so, good question. We only scratched the surface of that, I would say. It seems to me that a larger fermentation tank may remain more cost effective than a scaled hydrolysis process. I'd love to see a cost breakdown of what hydrolysates cost per kilogram at the moment, instead of what's required for cost parity, and to what extent this cost can be brought down with technical innovation. Yeah, these are good questions too, highly relevant. Again, we sort of scratched the surface on this in the analysis. So some people in the interviews gave us some indication of what hydrolysates cost today, but it's hard to really come to any specific conclusion because the hydrolysate market for cell culture is still relatively small. So it's not necessarily true that those prices might be indicative of what they could be produced at. Again, I think it's sort of like an immature area of development, perhaps, compared to some of the other media inputs that are used. And so we didn't spend a lot of additional time trying to source and dissect the technoeconomics of hydrolysate development. But again, that's great idea for future work, is to build some TEA models of hydrolysate processes using different scales methods, etc. We thought it would be valuable to provide a cost range target for people who are developing processes to know, essentially what they need to reach for in order for those for people to even consider using them from a cost perspective. Okay, Marie, I'll switch over to you for this question.
Awesome. Yeah. So someone asked that we mentioned that our price analysis was done with quotes for 100 kilograms of amino acid. And can we estimate how the price would decrease if we bought 1,000 kilograms or 10,000 kilograms? So I wish that we could. We really wanted to stick to real world quotes when we did this analysis. So we didn't publish any estimates around what it could be at larger volumes. I'd say we don't have an answer for you there. What I could recommend is if you took a look at the projected annual production, which again, low confidence, but if you took a look at that in the report and kind of identified some of the amino acids that are produced on a much larger scale, that might be able to give you a better idea of how low things can go when the scale is or the demand is increased. But there is one big caveat there, and that is that some amino acids are going to be harder or easier to produce than others. So I would also recommend looking into potential challenges or hurdles associated with the amino acids that you're interested in scaling.
Yeah, it's a good question. Obviously, if the industry does scale rapidly, then there will be higher purchase volumes associated with that that could come at, some lower costs. But yeah, we didn't really get to that point to being able to uncover that. I think partially because even the amino acid suppliers, they want to see the demand first before they start offering prices to people in the future. That's part of it. And I would say one of the other considerations, based on some of the interviews, is that some of the logistics with respect to shipping, like the type of container that you're using, these can also have sort of additive effects in decreasing cost over time. And there's maybe thresholds of volumes that will demand different types of shipping containers and logistics and management and that sort of thing. And so those are perhaps the areas that future studies can dive deeper into to really understand the smaller pieces of the puzzle that would need to be solved to further reduce costs, especially from like the supply chain management point of view, which we didn't really go too deep into in this analysis. Question from Netta. Do you plan to update the Delft LCA with this data? So there are no current plans for GFI to fund updated cultivated meat LCA work. It is something that I would love to prioritize. And if anyone has a couple spare $1 million or so, let me know and we'll prioritize that and get it to the top of the list. But yeah, there's not currently any plans. But I think, there's a lot of people obviously now that have been brought into the ecosystem that can lead these sorts of studies and build on them. And so hopefully taking some of the information that we included in this report, as well as some of the other studies that have been published, together would be able to provide a reasonable update, let's say, of an LCA study that is even more representative of commercial production. However, I do think that, as I mentioned, before that really gets done, first order of business is to, start to make sure that those inventory databases are fulfilled with real world data that is relevant for the media inputs being used today. I know there's a consortium of researchers that actually just published a guide on LCAs for cultivated meat that talk about this. And so would encourage people to look at that review paper that talks about some of the issues that, issues and data gaps that need to be filled maybe before people embark on these new studies. Okay, a couple more questions and keep them coming if you'd like. We're happy to stay on. Question is, in the hydrolysate blending approach, do you think hydrolysates could ever be produced cheaply enough to compete with food-grade amino acids, given that the expensive amino acids are already being supplemented separately? So as I mentioned, we got some information on costs, and I would say there's an overlap. So some people told us that hydrolysates could be purchased for um, prices that are within the competitive window that we published in the report. But again, we don't have any great level of confidence or validation for this. Um, the hypothesis, of course, is that if you're using something that is like a side stream or is produced in massive scales today with existing supply chains in the food system, commodity crop, that material is really pretty inexpensive to obtain. And so I think we just really need to understand, yeah, can we develop processes that will result in, consistent, reliable, high confidence inputs for cultivated meat? I guess, I don't have the answer to that. And if you talk to different people in the industry, some people will say, yes, of course we can. And then other people will say, no, I don't think we can. And so I think it's an open question that the research community will need to solve, I would say. Marie, do you have any thoughts on that? I know you maybe have worked with some of these hydrolysates yourself.
I mean, I'm ever the optimist. I'm very excited about the promise of hydrolysates, but it'll be a challenge in order to, you know, a challenge that I think that the industry is very capable of addressing. But it's not going to be easy.
Yeah. Um, okay, a couple more questions. Do you see a risk of negative perception by the consumer and public opinion if synthetic amino acids will be a major input, at least for the years ahead, until hydrolysates are manageable? Um, it's an interesting question. I suppose if you call them synthetic, then that might, lead to communication errors. So I guess it's, perhaps how you frame this. I assume there's no people on this call that want to harm the industry. But I mean, one of the things is, you can think of these being produced in bacteria. So people can come up with stories about how you're using bacteria to create things that are used synthetically in the media. So I think it's really like most things with cultivated meat and new food technology in general is, being able to educate the public, ensure that they have accurate information about how these inputs are actually produced, ensure that people understand that it's not really scary. It's highly similar to how a lot of food ingredients are produced today. Vitamins, is just one example. And so hopefully that will sort of normalize things and not create a negative consumer perception. But it's an interesting question and something that I don't particularly consider myself an expert on. So I leave that to some other people to maybe answer better. Question from Bert. Hi, Bert. Thanks for attending. Are the estimated quantities of hydrolysates required for the CM industry have any appreciable impact on the current supply of the source crude protein? What percent of total harvest and production would be needed for cultivated meat? Yeah, so I think those types of things would be interesting to study, especially if you get a sense of what raw materials could actually be used in cultivated meat production. So I think it sort of starts with narrowing down on, creating hydrolysates that work in a variety of use cases, not just like a single cell type for a single process, but something that could perhaps be more generalizable as a input into media. Then that would enable you to sort of do some of these calculations on, comparing the needs of the industry versus the current production volumes and how that's divvied up between animal feed and, conventional food, et cetera. I guess we can, I would note that we published 2 years ago an analysis called side streams. The story? Okay, it's being dropped in the chat. So yeah, we look at some side streams from commodity crops, and in that study, there's volume data. Um, I wasn't part of that work, but if I recall this being discussed, the the amount of volume that's produced with these commodity crops is just so large compared to what the cultivated meat industry would need anytime soon that it's basically like negligible for a very long time. And so I don't think it's something that we necessarily have to think about in the short term. But if we do start approaching millions of metric tons of production of cultivated meat, then those types of questions I think will be important to flush out better. Marie, do you want to comment on this last question around glucose?
Sure. So we had a question if we've made similar cost analysis studies for glucose and if we have a rough idea of the cost of glucose when bought in bulk. No. However, we do supply information around how much glucose is in our FSF4 media formulation. So if anyone is interested, they can certainly perform the similar calculations that we did for amino acids, but use the glucose amount and get an estimate of how much glucose we might need for 250 kilotons of cultivated meat production per year. But no, we were not able to include glucose in this study, but it's a great question and definitely something worth considering.
Yeah, I'll just add that when we go in the report, if you go to the section on the amino acid costs, we summarize a little bit of some of the existing techno-economic studies that have done. So people have tried to source basically individual costs for all of the major media components. Of course, they are using probably not the most accurate amino acid cost data, but nevertheless, that people have done this sort of mapping for total media. And generally what the studies suggest, I would say in summary, is that the cost drivers in media amount to proteins and growth factors and then amino acids. And the glucose, vitamins, salts, etc., make up, probably more or less, at least less than 20% of the media. If you have like no proteins in the media, then it's probably going to be even less. So it will be dependent on your media composition, but suffice to say, glucose, salts, vitamins, and most of the other basal media components make up a much smaller fraction of total media costs. But we do talk about in the report how, essentially what we hope people would do is take this updated pricing information and then do this sort of total media cost analysis with numbers that they can source, with food grade inputs for all of the other media components. And it is worth mentioning that, if you look on supplier sites like Alibaba for food grade amino acids, they actually aligned pretty nicely with the real world quotes that we obtained. That doesn't mean that people buy directly from Alibaba because, as was mentioned, the suppliers, there's differences in how they do maybe purification and impurities that are present, etc. So it doesn't necessarily mean that they'll perform well, but based on the information that we gathered for amino acids, you can get a pretty rough estimate, I think, that is, maybe fairly medium confidence if you just look on websites like Alibaba, madeinchina.com for food grade inputs. That will put you in a decent ballpark for cost. Hopefully people don't whip me for saying that. And then, so can this unavailability of the amino acid be considered a risk or technical barrier to the cultivated meat sector? I'm not sure if I understand that question. Maybe they could elaborate if you want to ask something more specific, unless Marie, you have a good intuition of what is being asked.
I mean, I think we did mention that if nothing was to be done, that it would eventually become a risk, just based off of what we found in terms of global supply. But I guess the plan is to be, forward thinking about this and make sure that we do everything that we need to in order to have enough amino acids by the time that we scale. Uh, Looks like we're
Yeah,
getting a little bit. Mhm.
I think it's important to sort of mention that that we're still a fairly long way away from 250,000 tons of production annually. So this is very much a forward facing analysis. There's not some general crisis of amino acid supply today, but really the point is that, if a lot of facilities get built successfully and scale up successfully, then you could have quite a rapid increase in the demand for amino acids. There's also other industries that use amino acids that, could be in competition to some degree. And so we wanted to really create awareness of the ones that could be focal points for the industry that could cause potential future problems, while also indicating, some of the longer term R&D opportunities for the field to focus on. Are we aware of any North American companies manufacturing AAs at scale? Yes. I guess you could probably ask the AI that question, and it would give you some lists. I don't feel the need to necessarily read out companies based in different locations. I think a lot of the companies that make amino acid suppliers are pretty large companies that might have sites at different locations around the world. And how those sites are utilized maybe depends on what type of ingredients that they're actually using or producing with their fermentation capacity. And so, yeah, I think that information can be found fairly readily. Another question on fermentation capacity is, do you see the existing shortage of fermentation capacity inhibit the growth of amino acid industries? Good question. I mean, we had some conversations, obviously, with amino acid suppliers and companies that source amino acids. I wouldn't necessarily say that I got the impression that people were worried about fermentation capacity. We do know there is still quite a bit of unused fermentation capacity, but maybe that is rapidly filled up by other alternative proteins needs in the future. Some of those just haven't necessarily materialized yet. So GFI has a separate report on fermentation capacity, maybe 2 or 3 reports that are relevant for this question. If my colleagues could drop them in the chat. But we did map out fermentation capacity and also potential for fermentation manufacturing infrastructure, particularly in Asia, where a lot of the capacities maybe exist today or is expected to increase in the future. And so, we have some other reports that might provide better answers to that question. But at least from the amino acids standpoint, it didn't seem like a pressing need at this time. Thanks, Amanda, for dropping that in the chat. Alrighty, well, thanks everyone for attending and asking your questions. We'd be really happy to hear your feedback. You can email me or Marie with comments, questions, tell us what you liked, what you didn't. If we got anything wrong, we'd love to know. But in the meantime, thank you so much for attending, and we'll be in touch. Bye.